In this section, we'll see the baseline proposal on performance objective for Campylobacter, Salmonella, Listeria monocytogenes, and VTEC in meat products. And we'll see how we define sampling plan to verify the compliance of meat lots to such performance objective. I'm Alessandra De Cesare, and I will present this result on behalf of the Work Package 4 of the baseline project. The approach we are going to suggest can be applied to different food-other combinations, keeping in mind that one performance objective must be associated to one food-other combination. The performance objective defines the maximum frequency or concentration of a hazard, in our case a biological hazard, in a food lot in a specific step in the food chain before the time of consumption. This maximum frequency or concentration contribute or provide the food safety objective and the appropriate level of protection. The food safety objective is the maximum frequency or concentration of the biological hazard in the food matrix at the time of consumption. So it's the same concept than the performance objective, but in another step of the food chain. The appropriate level of human protection in terms of food safety is the maximum number of a specific foodborne zoonosis a government deems appropriate in terms of public health. So, for instance, maximum number of Campylobacteriosis per year within the European Union, maximum number of Salmonellosis per year within the European Union. These figures show that the performance objective as well as the food safety objective are risk-based metrics. So even when the performance objective as a quantitative value is not a microbiological criteria. In any case, we worked on the definition of performance objective for food-other combination for which microbiological criteria are currently not included in the European regulations. The microbiological criteria define the acceptability of a product or a food lot based on testing of n sample units, in this case 5. The performance objective accounts for the distribution of prevalence and concentration of a biological hazard in a product or in a food lot. So basically, the definition of the performance objective might help the food producer to predict if, according to the distribution of prevalence or concentration of the biological hazard in their products, they will be accepted 100% of the time, like in this case, because the mean prevalence or concentration of the biological hazard in their products is low in comparison to the limit established in the regulations, for instance, 2 log colony forming unit per gram, or to predict if their products will be rejected 50% of the time because the mean of the prevalence or concentration of their hazard in the products is too close to the limit, the microbiological limit for that hazard in the regulations. What happened now with the microbiological criteria is that n sample units are tested, for instance 5. If C is 0, means that any sample unit might be out of specification. If only one is out of specification, the lot is wide draw, without a clear correlation with the risk, the real risk that that lot poses for human health. So it should be clear at this time that the performance objective and the microbiological criteria are both safety criteria, but they are based on different concepts. The performance objective are clear targets the food producers might try to achieve in order to contribute to the achievement of the appropriate level of protection. But the food producers are not really interested in the risk assessment models showing this correlation. So if we say to the food producer, you should try to achieve a maximum concentration of Campylobacter on broiler carcasses of 10 colony forming units per gram. This is a clear target for them. It's not really important for them the risk assessment model showing how the achievement of this level of Campylobacter on carcasses can be linked to a reasonable or an acceptable number of Campylobacteriosis per year. So let's see how we define the performance objective in our products for our specific food other combination. And let's start with Campylobacter in broiler carcasses. To come out with the performance objective for Campylobacter in broiler carcasses, we took the data collected by the European Food Safety Authority in Europe in 2008 regarding Campylobacter in broiler batches and on carcasses. During this survey, 561 slaughterhouses were included in the sampling, and more than 10,000 broiler batches were tested as part of this survey. From all these group of anonymized data, we took the data regarding 10,162 carcasses belonging to 3,400 batches collected in 25 different countries. In the European survey, they tested prevalence of Campylobacter colonized batches and prevalence and counts of Campylobacter on broiler carcasses. For our purpose, we just took those data regarding prevalence and counts of Campylobacter on broiler carcasses after chilling, because that is the point where the performance objective might be set. So regarding our results, when we speak about prevalence, we mean positive neck and breast skin samples obtained by detection or enumeration over the total number of samples tested. When we speak about counts, we speak about number of colony forming units of Campylobacter on carcasses. In this table, we summarize the risk factors impacting prevalence and concentration of Campylobacter on carcasses, which are slaughterhouse capacity, more or less than 10 to the 7 broiler processed per year, application or not of thinning, which is a depopulation procedure, time between carcass collection and carcass testing in the laboratory, and concentration of Campylobacter on carcasses. Here you see the number of data we analyzed in relation to each risk factor. The number of positive by detection or enumeration for each set of data, the number of negative for each set of data, and the number of sensor data, which are those data, those samples which were positive by detection but negative by enumeration. Then you see the concentration of Campylobacter on carcasses estimated using the maximum likelihood estimation method under the detection limit of the microbiological method, corresponding to 10 colony forming units per gram, assuming a sample size of 10 grams. These concentrations, you can see that range between 2 colony forming units in 10 grams up to 3 colony forming units in 100 grams. Then you see the mean and standard deviation of the distribution of Campylobacter concentration on carcasses. And this mean were taken as initial contamination level of Campylobacter after chilling. Based on knowledge of log normal distributions associated to the different risk factors, we calculated the performance objective, one for each risk factor. And you can see that the performance objective were defined as more than 99% of sample units tested with a concentration of Campylobacter lower than a value ranging between 2.5 and 4 log colony forming unit per gram. Then you see the microbiological limit as derived by the performance objective and the sampling options in terms of N and C which can be applied to verify the compliance to the different performance objectives. So if we take the first the first performance objective, more than 99% of the sample units with a concentration of Campylobacter lower than 3 log colony forming unit per gram, the sampling options are to analyze 5 sample units and 1 can be out of specification. So one can have a concentration of Campylobacter higher than 3 log, or to test 3 sample units and any sample unit can be out of specification, or to test 7 sample units and 2 can be out of specification. And then you see the other sampling options to be applied when the other risk factors occur. Let's move now to the second food and third actually food other combination, Listeria monocytogenes and Salmonella in pork cuts. In this case, any official data set and complete data set was available. So what we have done is to select a kind of pork cut and test this kind of pork cut in 10 different lots of meat products produced along a 1-year period within the same facility. In this pork cut, regarding Listeria monocytogenes, we tested both prevalence and concentration. Regarding Salmonella, we tested only prevalence. So here you see a scheme of what we have done. We tested our samples, which were this kind of pork cuts packaged under vacuum with the data logger inside to follow the time temperature shift along the product shelf life. So we tested our sample immediately after packaging at day 0 of the shelf life. After transport of this kind of product to a retail shop and storage in the retail cabinet at 4 degrees up to the day 4 or 5 of the product shelf life, and then after transport of 45 minutes at car temperature, and final storage in what we simulated as consumer refrigerators, set at 6 and 14 Celsius degree. This final storage was done up to the day 7, which was the last day of the product shelf life. These 2 temperatures were selected because according to a survey in the refrigerator of the consumers living in the area where the study was performed, 6 is the average refrigeration temperature and 14 degrees is the average abuse temperature. So what you see here are the beta distribution of the prevalence values of Listeria monocytogenes under the 4 different scenarios that we tested. S1 is after packaging, S2 is after storage at retail, S3 is after final storage at 6 degrees, S4 is after final storage at 14 degrees. What we basically have done is to take the 15th percentile of these distributions to derive the performance objective as maximum percentage of contaminated units in the lot. So we have seen that the performance objective for Listeria monocytogenes regarding the prevalence value increased between 40% after packaging up to more than 67% after final storage by consumers. If we want to derive the number of samples to test in order to verify the compliance of the food lot to this performance objective, we can apply the binomial distribution and 90% of confidence level and this formula, where P is the performance objective. So you see that the number of samples to test in order to verify the compliance to these different performance objectives decrease between 6 after packaging to 3 after final storage by consumers. If we look at the concentration data, because as I said for Listeria monocytogenes we tested both prevalence and concentration, you see that the concentration of Listeria monocytogenes in the pork cuts after packaging was around 2 log colony forming unit per gram. And this concentration, of course, increased during storage, especially at 14 degrees, when, of course, the pathogen had the chance to grow. So after final storage at 14 degrees, this concentration increased more than 1 log. Here you see the percentages of sample which were positive using enumeration. And this percentage increased between 5 after packaging up to 39 after final storage at 14 degrees. If we applied the maximum likelihood estimation method to the left sensor data, so again to the data which were, to the sample which were positive by detection but negative by enumeration, these percentages generally increase, except for the sample collected after storage at retail. Now, applying the maximum likelihood estimation method to the left sensor data, they were fitted to log normal distribution for Listeria monocytogenes concentration under the 4 different scenarios. And here you see the mean and standard deviation of the distribution of the concentration value. What we have done is also to check the variability associated to both mean and standard deviation, performing a bootstrapping analysis with 1,000 interactions. And this table shows you that the variability was very low, so the data were representative. This graph tells also you that the concentration of Listeria monocytogenes under the detection limit of the ISO method that we applied for the quantification of Listeria monocytogenes in this kind of products was close to the limit of detection. So this concentration of Listeria monocytogenes ranged between 7 colony forming unit in 10 gram after storage at retail to 7 colony forming unit per gram after final storage at 14 degrees. So you see that we are not so far from the detection limit of the ISO method, which is 10 colony forming unit per gram. This graph shows you the representation of the density functions of the log normal distributions fitted using the maximum likelihood estimation method. This graph is important because if this is the microbiological limit for Listeria monocytogenes in ready-to-eat products, calculating the area of the distribution above this limit, you can verify the percentages of samples not compliant with this microbiological limit. In other words, if we calculate the area of the distribution above this limit, so these different areas for the different scenarios, we can understand that 4% of the sample collected after packaging and after storage at retail are not compliant with the microbiological limit. This percentage increased to 8% after storage at 6 degrees and to 21% after final storage at 14 degrees. Regarding the definition of the performance objective for Salmonella, as I said before, we estimated the prevalence of Salmonella under the 4 different scenarios in the 10 lot of pork cuts. So again, here you see the beta distribution of the prevalence values under the same scenarios. So S1 after packaging, S2 after retail, storage at retail, S3 after final storage at 6 degrees, S4 after final storage at 14 degrees. Again, we took the 15th percentile of these distributions as performance objective. For Salmonella, you see that the performance objective decreased between 46% after storage at retail up to 26% after final storage at 14 degrees. In this case, you see that the number of samples to test in order to verify the compliance to this performance objective is different. Again, has been calculated using the binomial distribution and 95% of confidence level. So the number of samples to test in order to verify the compliance to the performance objective after storage at retail is 5, but is 10 if you want to verify the compliance to the performance objective established for products after final storage at abuse temperature. Let's move now to the calculation of the performance objective for VTEC in ground beef. For VTEC, we addressed 4 of the most relevant serotypes in terms of public health. For VTEC in ground beef, a comprehensive risk assessment model has been developed in Ireland. And here you see the flow diagram showing all the processing steps included in the risk assessment model. This flow diagram shows you that for a number of reasons I'm not going to explain now, trimming can be identified as the step where to put the performance objective. The performance objective for VTEC in ground beef was estimated with reference to a theoretical food safety objective value corresponding to minus 4.58 log colony forming unit per gram. This number corresponds to 3 colony forming units in 100 kg and has been estimated, as I said, is a theoretical value, taking into account the total number of VTEC human cases in the European Union in 2011 and assuming that the vulnerable consumer to VTEC follow an exponential dose response model. So, to make this graph simple, the performance objective was estimated using the intersection between the concentration of VTEC at trimming and the theoretical food safety objective value. So the performance objective estimated for the serotype 0157 and 026 are 6 colony forming units per gram. The performance objective estimated for the serotype 0103 and 0145 are 40 colony forming units per gram. So for VTEC, we estimated different performance objectives for the different serotypes. But this makes sense because these different serotypes have different infective doses for humans. So to come to the conclusion for this section, we have seen that in order to calculate performance objective, you need to have representative data describing prevalence and concentration of a biological other in a food product. This data should account for the intra-lot and inter-lot variability and should account for uncertainty. We also have seen that in order to come out with the number of samples to test to verify the compliance to the performance objective, you can use the binomial distribution and 95% of confidence level. This approach provides the food producer with the number of samples to test in order to have 95% of probability to detect a positive sample if it is there, without asking them to test an unreasonable number of samples. Of course, we selected 95% of confidence level. For risk, from a risk assessment point of view, different percentile can be selected, but this is a choice of the risk manager. We also have seen that in order to verify and to define a performance objective, the fate of the other in the food lot must be known up to consumption. And this is very important because the performance objective is a risk assessment metric. The value of the performance objective and also the value of the food safety objective should be linked to the probability of that biological other to cause an infection in humans. And this link can be done only using risk assessment model. So the key message is that more risk assessment model for specific food other combination should be developed in the near future. And in this module, particular attention should be set for the exposure assessment and the dose response. We hope this section was useful for you. If you need further information, please contact us. Thank you.